Hey everyone, I'm Kevin Tan. I'm a first-year grad student with Matt Lieberman and Carolyn Parkinson, and I'll be presenting the first intracranial electrophysiology study of mentalizing. Mentalizing, the ability to consider the mental states of oneself and others, is one of the most important human abilities, and one that has been studied repeatedly in social neuroscience. There are literally hundreds of fMRI studies on mentalizing, and whether you look at individual studies, meta-analyses, or Neuroscience, the results are remarkably consistent. The default network regions shown here are consistently recruited by countless forms of mentalizing, yet it's been difficult for us to go beyond this network level identification. One reason for this may be that the unit of analysis in fMRI, the TR, is usually 2 seconds, but we know that neurocognitive processes occur at millisecond timescales. Thus, little is known about the neural dynamics under the hood of a TR, trial, or block in fMRI studies of mentalizing. Moreover, the same bold result could stem from many different combinations of the magnitude and timing of underlying neuronal activity. For example, higher bold activity for others versus self doesn't have to arise from differences in the magnitude of neuronal activity, but instead differences in the duration of neuronal activity, or any combination of these two. The green lines in both toy examples representing the other condition produce the same bold result in a TR despite having drastically different time courses. And while this is an oversimplification of the relationship between bold and neuronal activity, I hope this illustrates that there's critical temporal information not captured within a TR. So, taken together, it's apparent that high spatial and temporal resolution is required for a truly incisive understanding of the neurocognitive underpinnings of mentalizing or any other cognitive construct. To probe the social brain with millimeter spatial resolution and millisecond temporal resolution, we conducted the first electrocorticography study of mentalizing in collaboration with the Parvizi lab at Stanford Neurology. ECOG is an invasive technique that involves surgically implanting electrodes onto the cortical surface or deep within cortical and subcortical tissues. In humans, ECOG is typically reserved to treat patients with drug-resistant epilepsy, but some of these patients kindly volunteer to do studies like ours. Today, I'll be focusing on the high-frequency broadband spectrum, which reflects the spiking rate of neuronal populations immediately adjacent to an electrode. Unlike bold, HFB is a direct measure of neuronal activity. Don't worry, no more gory pictures from here on out. Here's the total electrode coverage for our current cohort of subjects. In ECOG, each subject has a different montage of electrodes, but as you can see here, near whole brain coverage can be achieved by using a cohort of subjects. But do keep in mind that each region of the brain has a different mix of subjects. We parcellated the brain into 5 a priori ROIs for our analyses. Blue is visual cortex, green is TPJ, yellow is PCC and precuneus, which I'll refer to as just PCC for short, pink is dmPFC, and purple is vmPFC. In our task, we compared 2 mentalizing trials focused on either the self or others. Participants judged whether they or a close other possessed particular psychological traits. We also had non-mentalizing trial types, such as math, episodic memory, semantic memory, and cued rest. However, I'll just be focusing on mentalizing for now. I'm going to be talking about the onsets, peaks, and offsets of the HFB signal, so I wanted to orient you by showing what the data looks like within a single electrode, in this case, a visual cortex electrode. These time courses represent mean differences in HFB activity relative to the pre-stimulus baseline within each trial. Solid lines represent significant differences from baseline, thresholded at p under 0.05 FDR corrected. Dotted lines represent non-significant differences. I'll be examining 3 important metrics in these time courses. The first metric is the onset, or the time at which significance is first evoked. The second metric is the peak, or the time and magnitude at which the maximum difference from baseline is evoked. The third metric is the offset, or the time at which significance is last evoked. Here are latency metrics averaged from ROIs, or from electrodes within each ROI. The left edge of the bars represent onsets, the right edge of the bars represent offsets, and the diamonds within the bars represent peaks. The onsets reveal a clear sequence of activity across ROIs, implying a hierarchy of processing in mentalizing. Activation begins in visual cortex, suggesting that it underlies the lowest level of processing as you'd expect. Then comes on TPJ and PCC, roughly at the same time, and after that, dmPFC, and lastly, vmPFC, which appears to sit at the top of the hierarchy. Now, this vmPFC result may surprise you, given that this isn't a particularly affective mentalizing task. However, we've recently published a neurosynth analysis with Megan, actually, showing that some parts of vmPFC are more associated with social cognition than affect, while some parts of vmPFC are associated with the situational and schematic processing that contribute to many types of abstract high-level cognition. But the bigger point here is that just because the region or voxel shows higher bold activation for a certain psychological domain, affect in the case of vmPFC, that doesn't necessarily mean that that region is only specific to that domain. In fact, non-specificity seems to be a recurring theme in the mentalizing regions I've looked at in this ECOG data. Difference between self and other mentalizing show up in the peaks and offsets of these ROIs. Other evokes later peaks and offsets, but only at high levels of processing. This may suggest that other focused mentalizing takes longer at higher levels of processing and that these timing differences are additive at succeeding steps of processing. Now, to take a step back and loop everything together, using fMRI has allowed us to identify a network of regions that underlie mentalizing. However, it's been difficult for us to parse the relative functions of each region. Using ECOG, there's a clear sequence of activation across mentalizing regions, with parietal regions coming on earlier, followed by medial frontal regions, and towards the end, we pretty much only see vmPFC. Now, we have ideas about what this sequence implies, and I bet you might have some ideas too. But the real point here is that data like this can help us see and think differently about the mentalizing system, allowing us to test new ideas that may help reveal the relative functions of each region and the interdependencies between them. So while there's an overall similarity between self and other mentalizing, there are also differences between them. Other mentalizing evokes strong activity in dmPFC at 750 milliseconds and beyond. In contrast, self-mentalizing evokes earlier and shorter activity. So rather than this prototype of bold differences that we get from fMRI, ECOG reveals that the real dynamic in dmPFC looks more like this, where self-other differences arise mostly from the duration of neural activity rather than the magnitude of neural activity. These results are consistent with the idea that self and other mentalizing rely on roughly the same processes, but because we know ourselves better, we're more efficient at self-mentalizing. In other words, the difference between self and other might be more due to computational load, such as social working memory, rather than what mentalizing type dmPFC cares about. So in conclusion, these results are a very preliminary first step. We have a ton of additional work to do with this data set, and we're already planning our next study. I do hope that this talk gave you a sense of ECOG's promise as a complement to our current techniques in social neuroscience, helping us refine and advance our understanding of the neurocognitive systems that help make us social. Now, I don't have time to get into all the other interesting results in this data, such as TPJ producing a lot more HFB activity for episodic memory compared to self or other mentalizing. Fortunately, I'll be presenting again at Poster B1 at 5:30 today, so you can ask me about this or anything else at the poster session. Maybe Dr. Canwisher can tell me why I'm wrong. I'd like to give many thanks to my collaborators, funders, and to you for your time. And again, I'll be at Poster B1 if you want to see more data. Thank you so much.